Hi, thank you very much for the invitation. In particular, I'd like to thank Dr. Curtis Rempel for his invitation from the Canola Council of Canada. So today I'm going to talk to you about omega-3 fatty acids and the evidence for their usefulness in human health. Before we get into the talk, here are some disclosures. I've received consultation fees, honoraria, or travel support from the following entities, and I am currently funded by the following funders. So to start off, I'm going to get into some data from others just to set the stage for what's currently known about omega-3s and how they impact human health, and then show you a little bit of work that we're doing in my lab with my collaborators. But in consultation with Dr. Rempel about this talk, we decided that we should probably do a little primer on fats, and in particular, essential fatty acids, and why they are important in our diet and why they may be pointing towards ensuring adequate intakes to prevent chronic disease development and these sorts of problems that we develop over the course of our life. But if we come back and just look at dietary fat as a whole, we know that, or most people know that fat is probably the most energy dense nutrient in our diet. And it is mainly used by our bodies for that purpose, for the utilization of energy to produce ATP for our cells. There is also the importance of fat in our diet because it's also a carrier for the fat-soluble vitamins A, D, E, and K. And without proper amount of fat in your diet, you wouldn't be able to absorb those important vitamins. It also provides the essential fatty acids, and the essential fatty acids are those fatty acids which our bodies cannot make. Therefore, they are dietarily essential. But for a lot of people, food is not very palatable unless it contains some fat. And that's characteristic for many individuals, partly because fat actually dissolves many of the flavor compounds that we appreciate when we consume food, as well as creates characteristic mouthfeels, all of which go to the hedonic pleasure component of consuming food. So what are essential fatty acids? Well, we have a number of fatty acids in our body that we get from our diet and we create ourselves. We have the capacity to make them. For example, this is stearic acid right here. It's 18 carbons long and it has no double bond, therefore no unsaturation. Shorthand notation for that is 18-0. And then here you see oleic acid, again, another fatty acid we can make substantially in our body. And it's a monounsaturated fatty acid. So you see here in the carbon skeleton, 18 carbons with a single double bond at the carbon 9 and 10 position. We have no problem making these fatty acids. The problem is, once we get to this stage, we can't do the next step, desaturating the fatty acid further. Animals can insert as many double bonds as they need from in this area of the chain. However, they can't do it here. And therefore, these two fatty acids are essential in our diet. And these are the 18 carbon omega-6 fatty acid, linoleic acid, and the 18 carbon omega-3 fatty acid, linolenic acid, alpha linolenic acid. Once we consume these in our diet, our body has the machinery, and by machinery I mean the enzymatic capacity, to create all the other fatty acids required through steps of either desaturation, introducing a new double bond, or elongating the fatty acid, making it longer than 18 carbons. And you can see here from this diagram, it's rather simplistic, but it illustrates that both the omega-6 and the omega-3 fatty acids use the same enzymatic machinery to produce very different longer chain polyunsaturated fatty acids. If you look here, you proceed through to gamma linolenic acid, to arachidonic acid, which some of you may have heard of, all the way down to an omega-6 docosapentanoic acid. On the omega-3 side, you see that alpha linolenic acid is the starting point, and you can proceed through to eicosapentanoic acid or docosahexanoic acid. These are very long chain polyunsaturated fatty acids important to our health. So in terms of essentiality, these two fatty acids are the essential fatty acids for humans. You can obtain linoleic acid, the omega-6, from pretty much any vegetable oil. And in most countries, and I would probably go out on a limb and say pretty much globally, essential fatty acid deficiency is rare and usually only seen in clinical situations or situations where there's endemic protein energy malnutrition. It's not a common condition seen as it would have been in hundreds of years ago when fat sources were monotonous, plant-based sources and marine-based sources of fat weren't as ubiquitous as they are now. So there's very little, if any, deficiency seen unless there's a clinical reason for that deficiency. Like I said, and that could be related to either not eating or being protein energy malnourished, or it could be related to some kind of metabolic disorder. When you look at the omega-3 fatty acids, some people will equate eicosapentaenoic acid and docosahexaenoic acid, the marine omega-3s, as being essential. And while they are important, they are not essential for most individuals. Just the alpha-linolenic acid, which is a plant-sourced omega-3. Once we have this fatty acid, we're able to produce eicosapentaenoic acid and docosahexaenoic acid. You can see that I do have listed here that it is conditionally essential, and I'll get into some reasons for that later, but humans can, in general, produce this fatty acid in most of their tissues, but it is at a low level. But that low level is at the level that we require, except for in certain circumstances. In terms of structure, this is how they look. So these would be the three main omega-3 fatty acids that you hear probably the most about, either in health or in scientific research and these sorts of things. And you can see the 18 carbon length alpha-linolenic, 20 carbons for eicosapentaenoic acid, and 22 carbons for docosahexaenoic acid, each with their double bonds, their corresponding double bonds. Now, that seems pretty straightforward, but when you get to the level of the consumer, there's a lot of confusion surrounding these products. I'm showing you here just a small selection of recent and one not so recent fish oil supplement. And I should back it up and not even say fish oil, because as you can see, two of these are not fish oil. So these are omega-3 or omega something fatty acid supplements. And you can see here, if you look over at this Nature Made one here on the left, it contains fish, flaxseed, safflower, and olive oils. But it's also listed as the triple omega, omega-3, 6, and 9. And like I said a minute ago, the omega-9 fatty acids, we make that in our body, and it's not essential. You don't require supplementation for that. It is also extremely ubiquitous across most fats and oils that we use in our diet. Omega-6 and omega-3, if it's not linoleic acid or alpha-linolenic acid, it's not essential, and our bodies can create them. Over here you see flaxseed oil. This is primarily, it's probably going to be 50 to 60% by weight, alpha-linolenic acid, because flaxseed is very high in that particular fatty acid. Here you see we have krill oil and Pacific salmon oil, and then a very old advertisement for cod liver oil. So these products are promoting the marine source, which are not essential fatty acids, but they are promoting them as supplementary use for health. You can see here it's actually listed on the package for the krill oil: arthritis relief, healthy heart, and healthy cholesterol levels. There's a few little points made here in this older moniker by Peter Mahler's cod liver oil. And there's probably some on this natural factors, omega factor container as well, but I'm only showing you the front here. You do see that it maintains cardiovascular health as listed here. So these are all health claims surrounding these supplements, but in terms of their essentiality, they're not actually providing you with a fatty acid that your body can't already create, provided you get enough alpha-linolenic acid. So what is the evidence for why is it emerging and so popular in the supplement industry, as well as in the health advertisement and promotion industry, that omega-3 fatty acids are essential to get in your diet in supplementary forms or in higher levels than you would have been concerned about previously? If you look through the literature or the clinical trials databases, there are a number of current supplementation trials going on or have recently finished that are looking at supplementation of omega-3 fatty acids, in particular the marine source fatty acids, looking at these for the treatment of various conditions and diseases, things like autism and depression, using them as secondary treatments or adjuvant treatments for cancer, attention deficit hyperactivity disorder, age-related macular degeneration, and the reduction of inflammation. So this is a pretty wide-ranging example or cross-section of the number of types of studies. And each one of these national trial registry codes is referring to a different study. You can see there's multiple studies for some of these conditions going on right now. There are probably more. If you took a look at clinicaltrials.gov and then search for omega-3 supplementation, you'd probably find a lot more than what I'm showing you here. So then what are the positive health effects of omega-3 fatty acids? And is it specifically marine fatty acids, marine omega-3s, or do we get the benefit from both the plant-based as well as the marine-based? How did it even start? That's another question. Where did this kind of push come from? If you look back in some of the older literature, there was probably one of the landmark studies for the promotion of omega-3 fatty acids, in particular marine fatty acids, for improving heart health or health in general, comes from a study from some Danish researchers back in the 1970s. These researchers were studying the diets and the health of the Greenland Inuit, which is part of Denmark. And they were comparing these diets, and then they got into some of these health indicators against the mainland Danes who live in Europe. And if you look at this very brief little table here, what this is showing you is one of the results that they found and reported in this Lancet article from the 1970s. They measured the type of fatty acid that was present in the phospholipids of the platelets of the individuals that they took blood from. They recognized that the Inuit have a high fish or high marine mammal and fish diet. And what they looked at was the composition of the platelets and the phospholipids in particular. And this gave them an indication of what the background diet's effect were on the whole body's distribution of these types of fatty acids. So AA refers to arachidonic acid, EPA is eicosapentaenoic acid, and DHA is docosahexaenoic acid. AA is one of the major, or arachidonic acid is one of the major omega-6 fatty acids in our body. You can see here that it was pretty close to a 1-to-1 ratio between these fatty acids for the Inuit, whereas the mainland Danes had a very stark difference in terms of their fatty acid distribution. You can see that it's about 40-to-1 for EPA to AA and about 22-to-1 for DHA to AA. Then they did this functional assessment, and this is called clotting time. And clotting time is an experiment where you take a drop of blood from an individual and put it on a microscope slide that's inverted. It's on an angle, and then you time it with a clock how long it takes for that blood to clot. Hence the name clotting time. It's pretty intuitive. And when they did this experiment, you can see that the Inuit had a 40% longer clot time result, 8 minutes on average versus 5 minutes for mainland Danes. So this was probably some of the earliest evidence that there's correlation, I won't say any causation at this point, but correlation between your diet, the omega-3 content of that diet, the omega-3 makeup of your plasma membranes and the fat in general in your body. And then when you look at the health outcomes in these different populations, the Greenland Inuit had a 10 times lower incidence of heart attack and stroke compared to the mainland Danes. They had lower cholesterol, LDL cholesterol, and triglycerides, and they also had higher HDL cholesterol. So all of these things, while nothing here, this is all cross-sectional, nothing here is pointing towards a mechanism for what's happening. This is all very much pointing towards a correlation between these marine fatty acids and improved heart health. Since that earlier study, more research has been done to determine what types of roles these very long-chain polyunsaturated fatty acids play in the human body. And there are a number of organs which are very rich in these fatty acids, and they serve very much functional purposes for normal health in these particular tissues. So, for example, if you look at the eye, if you look at your retina, DHA is the major polyunsaturated fatty acid found in the phospholipids in the discs of the rods in the retina, and as well in the synapses of the brain. So you can get an altered electroretinogram, which is a measurement of how the retina is functioning, and you get impaired visual clearness when DHA is replaced by docosahexaenoic acid, a different long chain polyunsaturated, and then as you can see, it's an omega-6. The rate of synthesis of DHA from alpha-linoleic acid can be limiting in some individuals, like a preterm infant. And so this was discovered probably later in the 1980s. And when this was identified, that's when you would see the advent of new formulas for preterm babies, which contained DHA and EPA to supplement these preterm infants who didn't seem to have the capacity to convert alpha-linoleic acid to these longer chain polyunsaturated omega-3s. So, but if you look at a term infant, the need for DHA can be met by the dietary alpha-linoleic acid. So this is just two examples by which you can see there's a physiological role to play, or there's a major physiological role for these polyunsaturated fatty acids, both omega-6 and omega-3, and there are situations where supplementing makes sense. So when you do supplement, it doesn't take much and it doesn't take long to see a very significant change in your overall fatty acid distribution across your body. So this is a study by Phil Calder from University of Southampton in the UK. And what he's showing you here is that when you supplement an individual at 2.1 grams of EPA and 1.1 grams of DHA per day for a week or 12 weeks, you get a shift in your fatty acid profile. What they did was measure the membrane fatty acids in mononuclear cells in healthy volunteers. And these volunteers were also consuming fish as well. So they had a baseline level of omega-3 fatty acid anyway because they were fish consumers. But then when you add the supplemental DHA and EPA, what you see is a shift downwards from the arachidonic, which is a major omega-6 fatty acid. So you're going from about just under 20% down to about 17.5% of the total fatty acids in these cells, in the membranes of these cells. But then when you look at the DHA over that same period, in a 1 week period, you go from about 0.5% to 1%, all the way up to about 3% for EPA and DHA in the membranes of those cells. What's interesting is when you look at it for the next 12 weeks, it doesn't change. It stays pretty much the same. It's slightly increasing, you can see there, but nothing significant. And the same for the change in the arachidonic acid. It doesn't decrease any further. So you can reach a saturation point. And while it's not on this particular graph, obviously if you stop this supplementation, you would expect over a number of days or weeks, you'd return to these baseline levels of EPA and DHA in the membranes of your cells. So we know that supplementation works, but there's an upper limit, there's a capacity to which most individuals will reach, and going beyond that level of supplementation won't have any impact on your whole body. The fate of those fatty acids that if you continue to supplement or you supplement at a higher level would be the same as any other fats. They would be burned up as fuel, metabolic fuel, to produce ATP in your cells through the process known as beta oxidation. Now, if you look over here at the positive health effects for omega-3s, you can see they're listed here. These have been established by numerous studies, and there are various systematic reviews on each of these. But they will decrease serum triglycerides, which is a major risk factor for cardiovascular disease. They have an antiarrhythmic effect. They will stabilize atherosclerotic plaques. They will have a blood pressure reduction effect. It's only modest, and it takes very high levels of supplementation to do this. And they have an anti-inflammatory effect, and some of this is suggested to be through the reduction of arachidonic acid levels. But again, if you look at the level of supplementation, it's above the level that was shown in that last graph. That we're up around 3 to 4 grams per day, and this is of specifically marine-based fatty acids. This is not ALA. So a quick case study just to show you how diet will impact your fatty acid levels, so the intake in your diet as opposed to supplementation, and how that looks in terms of your blood fatty acid levels. So this is from the EPIC. So this is the large cancer prospective cohort study that was done across a number of European countries. This is specifically looking at the Norfolk, UK data, so EPIC Norfolk cohort. And what it's looking at here is they took the individuals, these 2,600 men and 2,400 women. I'll show you the women in the next slide. And they broke them out into fish eaters. And you can see this is fish eaters, but they eat obviously meat as well because it's 2,000 of these individuals. Then they had meat eaters who ate no fish, vegetarians who ate no fish, and vegans who ate no fish. You can see the sample for vegans is very small and probably not very reliable. So you can probably, if you want, combine both of these just to get an idea of what these numbers will look like. Mean age was in the 60s. And then if you look at total omega-3 fatty acid intakes from diet, you can see that the fish eaters had the most, which is probably not surprising. That's intuitive. And these others had progressively less. And if you look at the vegans, this is important when we get to the next couple of slides. Look at the vegans, they're down, they're about 60% or 40% less than fish-eating omnivores that you see over here. If you specifically zone in on omega-3s and then look at DHA, in the diet, DHA is not a major fatty acid when you look at the total N3 PUFAs or omega-3 PUFAs that are being consumed. This is the data for the women, so 2,300 women, same kind of distribution. And then if you look at the fatty acids, slightly less than men in terms of intakes per day, but this is grams per day. Women tend to have smaller body weights, and most people eat towards what their current body weight is, so that's hence these lower numbers. Remember, this is dietary intake. And then so if you look at this, it's the same trend, decreasing towards the vegans, with the highest being the omnivores, who are also fish eaters. And same trend with the DHA percentage in the total PUFAs in the diet. Now, let's look at their blood fatty acid concentration. So this is telling you both what you're eating, but also what you're converting endogenously to from these fatty acids in your diet. And if you look here at the total omega-3s, this is again the males, the male data. You've got 360 micromole per liter of total omega-3s. But look at the DHA. It's higher than the ALA. It's higher than the EPA and DPA. It's 236. Okay, so it's significantly higher than if I just flip back to the diet for the men. You can see here that ALA is actually the predominant, making up upwards of 75% of the omega-3 in the diet for these men. But when you look at their status in their blood, you see that the DHA is actually much higher. And this trend is across the board for all of these types of fish eaters, meat eaters. Fish eaters seem to have a little bit more, but if you look at the variation here, none of these are significantly different from each other. These are all virtually the same. So this is giving you an indication of that the endogenous capacity, I'm showing you the women's data here now, is the same as that of the endogenous capacity to produce these very long chain polyunsaturated omega-3s, namely DHA and EPA, is met by the dietary requirement. You see the same trend here, much higher DHA level compared to ALA level in the blood plasma fatty acid concentrations. And this trend stays the same across the board. And interestingly, even though keep an eye on this variability here, it's quite variable, even a vegan has the capacity to produce this DHA at the same level as an omnivore. So this gives you an indication that it's not linked to diet, and it's more so linked to requirement of these types of fatty acids and your body's ability then to convert from the essential fatty acid ALA into these longer chain omega-3 PUFAs. So what does the data say about supplementing with omega-3s, and in particular the marine-based omega-3s? Well, two recently finished and very large supplementation studies were conducted. One is called the ASCEND trial, and this is a study of cardiovascular events in diabetes. This was conducted through the University of Oxford, and its primary outcome was to determine if supplementing with omega-3s from marine sources could reduce the incidence of myocardial infarction, stroke, ischemic attacks, or vascular death and this didn't include cerebral hemorrhage in this particular study. It was a 7.5 year prospective study, and it was registered at clinicaltrials.gov, and I've put the indicator there in case you want to go see some of the data that was reported in their clinical trials entry. The main outcome, though, if you don't want to read the paper, is that supplementing omega-3s, marine-based omega-3s, in the approximately 7,700 volunteers, 63% of which were male, this was a 2-arm study, there were no significant differences between the omega-3 supplemented or placebo group in the incidence of serious vascular events, all of which are listed above. So that's a heart attack, a stroke, a transient ischemic attack, or death from either of these. Here's a main exhibit of their data, and what you see here is the incidence, the percentage of patients in the entire cohort who had one of these events. So it was any of those events that I mentioned previously. And you can see as the study progressed through the years, they actually went out to 9 years, as you can see, there's virtually no difference in the rates between the placebo group or the group that received omega-3 fatty acids. So this was a high dose omega-3 fatty acid. I think it was a 1 gram total, and of that, about 75 or 80% of the fatty acids were DHA and EPA. The second study I'll show you, which from what I can tell based on the composition in the papers of the fatty acid supplements, it looks like they probably provided the same supplement. It's a commercially available one. And what you see in this study is looking at omega-3 supplementation as well as vitamin D supplementation. This is a 4-armed trial named VITAL, or the VITAL study, and it's linked to vitamin D and omega-3s. It was mainly run by researchers from Brigham and Women's Hospital in Boston, and they refer to themselves as the VITAL Research Group. The outcomes here were looking at cancer and cardiovascular disease development in a 5-year prospective trial. And so again, these individuals were provided with a marine-based omega-3 fatty acid supplement and followed up for 5 years. If you look at the 4 arms, they were all very well balanced, 6,500 participants in each arm. It was sex balanced, basically 50-50 male and female for this. What they did show was that, again, there were no statistical differences in cardiovascular events or deaths between the groups. But they did see a 28% reduction in total myocardial infarction for those who were consuming the omega-3 fatty acids. Here's another exhibit taken from their paper, their main publication on this. And you can see that, again, similar to the ASCEND trial, the rates are very much the same. So this is the cumulative incidence of cardiovascular disease events over the course of the study, follow-up for 6 years. You can see they're virtually the same, but you do see that those consuming the omega-3 fatty acids seem to be diverging from the placebo group. The rate is roughly the same as you continue on, and they saw no statistical difference. Because if you look over here on the side, we're looking at 3% here, so 3% of this 12,000 individual group, or sorry, 24,000 plus individual group. It's a very low number, and they're not statistically different from each other. And then if you look here at the myocardial infarction, where they said they have a 28% reduction, it looks very big here when you look at this, but remember the scale over here. This is about 1%, right? So this is the difference in terms of incidence in the group is less than 1% of the group itself, even though it is a 28% reduction, which must have showed as being statistically significant. So the evidence from the supplemental trials for marine-based omega-3s don't show any significant benefit to supplementing. And therefore, I think more work needs to be done in terms of targeting very specific situations or health situations towards these supplementation trials, as opposed to looking at them in the broader population where nothing seems to be jumping out in the data to suggest that there's any benefit to it. Now, let's change gears a little bit and look at the plant-based omega-3 fatty acid, alpha-linolenic acid, and how intakes here, this is dietary, not supplementary, how dietary intakes are affecting mortality and how it links to cardiovascular disease. There have been 2 very recent systematic reviews which have been looking at the prospective cohort data and clinical trials data These are both registered in the Prospero Systematic Review Registration System, and their numbers are here. You can look those up if you'd like. But both of these papers are published. First one is Chen et al. 2021, and this is looking at dietary intake and biomarkers of alpha-linolenic acid and mortality. And then the other one is Negishi et al. 2021 again, and you can see dietary intake and biomarkers of alpha-linolenic acid with all-cause cardiovascular and cancer mortality. Again, this is systematic review and meta-analysis. So what these studies demonstrate is that the preponderance of evidence from the studies that they included in their reviews show that with higher ALA intake, you get an inverse association with all-cause and cardiovascular disease-related mortality. Remember, this is dietary data. This is not from supplemental sources. Negishi et al. Showed something very similar. They had theirs broken out as all-cause cardiovascular disease-related as well as coronary heart disease-related mortalities, and they also showed an inverse relationship with ALA intake. So what both of these systematic reviews tell us is that the current state of the knowledge based on the trials that have been done or the cohorts that have been followed show that individuals consuming higher levels of ALA in their diet tend to have less risk of dying of a cardiovascular or coronary heart disease related event. And that includes all causes as well, which are not related to the heart. The biomarker data from Negishi et al. Was interesting as well because they showed that those individuals with higher blood ALA levels, so again, similar to what I showed you with Phil Calder's study with supplementation, as well as the Inuit, the Greenland Inuit versus mainland Dane study, these levels of fatty acids in the blood, if the ALA levels were higher, it correlated with a lower risk of CVD and CHD mortality. So these data really support higher intakes of ALA in the diet, not necessarily supplemental levels, but higher dietary levels, which is a very interesting finding given the more regularity that we see now that recommendations for healthy eating change in various jurisdictions, like in Canada and the US, for example. Now, another proviso on this data is that most of the studies in both of these systematic reviews were North American in nature, which means this might not be broadly applicable to other jurisdictions. It would be interesting if there could be some type of study done to compare cohorts that were studied in this way, dietary intakes of ALA, and then these diseases, but looking at them by jurisdiction as opposed to just looking at them based on the study designs as presented in these two studies. So with that, I'll shift gears slightly and we'll just talk about some of the mechanistic work we're doing in my lab. Myself and my collaborator, Dr. Rob Bertolo, have been working on a study that looks at omega-3 digestion, absorption, and tissue distribution. But we've been looking at it more so from the perspective of can we augment tissue uptake, basically, through dietary means. So if you're going to supplement in the particular case of the study I'm going to show you with fish oil, can you improve its uptake and distribution through the body by adding some additional phospholipid in the diet as well to try to shift that supplement towards being taken up as a phospholipid as opposed to a triglyceride? And I've got some illustrations here just so you can see that what we're looking at is when these get broken down in the diet, when they're being digested, these fatty acids at the SN1 and 3 position on the triglyceride, those are the ones that are actually cleaved off. And then they get absorbed. And once they're in the enterocyte, they get repackaged. Phospholipids get broken down in a similar way, except it's the SN2 position fatty acid that tends to be removed. And then the lysophospholipid is absorbed with a free fatty acid. And then what we were trying to do in this particular study was to see if pigs fed a very poor diet, poor in the sense that it's a Western diet, and our Western diet is based on the intakes in North America for salt, sugar, and saturated fat. And what we did was we designed a diet for these pigs that contained their background diet, but then put them in the 95th percentile for intakes for humans for salt, sugar, and saturated fat. This was the control diet, so this is just showing us what impact this type of diet has on the pigs. Then we had a Western diet plus fish oil. The fish oil was about 5% by weight of the diet, and that particular fish oil was 13% by weight DHA and EPA. Then we had a Western diet with the fish oil at the same rate, but then we added phosphatidylcholine in the form of soy lecithin. And the idea here was, like I said, to improve emulsification of the fat in the diet, as well as to hopefully push the reassembly of the fatty acids to the tags or to the phospholipid in the enterocytes, more so towards an increased repackaging of the DHA and EPA in the phospholipid form. These pigs were fed for 12 weeks, and we have two students who are working on this data. F.A. Obade has been working on the fatty acid uptake and distribution, and then we also had a master student who was really interested in looking at the preventative health effects of the fish oil in these pigs with the poor background diet. And so what we've seen so far is, well, first I'll give you a look at the diet. You can see this is just regular swine diet that they would consume if they were just being grown as normal pigs. Then we added the sugar, salt, and then the fats to get them to a high saturated fat level. And then we augmented the polyunsaturated fats with the fish oil and with the lecithin when we added it in, because the lecithin does contain some fatty acids, so we had to augment that. You can see our omega-6 to omega-3 ratio down here. 45 to 1 for the Western diet, 2 to 1 and 2.25 to 1 for the fish oil and Western diet. And they're pretty much the same in terms of caloric intake, the caloric density of these diets for the pigs. So this is some of FA's preliminary data. This is probably not the final version, so this is definitely unpublished data. We need to do some additional analysis, but right now, I'm just going to show you some of the trends we're seeing. What I'm showing you here is the liver and the heart, and we looked at the total saturated fatty acids, the polyunsaturated fatty acids, the N6 variety, and then the N3, and then I've shown you the EPA and DHA levels. And things were going in the right way in this study, the way we anticipated they would with the additional fish oil, the additional EPA and DHA, where you have the Western diet having a higher saturated fat level compared to the fish oil and PC group, as well as even though they all contained the same level of saturated fat, then you see the N6 PUFAs are higher in the Western diet compared to the fish oil groups. And then if you look here at the omega-3s, you can see they're much higher in the fish oil groups compared to the Western diet, in particular EPA and DHA. And so this is percent, so this is weight for weight, looking at the tissue fatty acid distribution. Heart looked pretty much the same, and as you can see over here, there's higher levels of omega-3 fatty acids present in the heart. We got some interesting data here that we're not entirely sure it's correct at this point. I'm showing you anyway just to give you an illustration of what we're doing in the lab, but you can see there's some anomalies there that we weren't expecting with the fish oil. Then if you look at the brain and the retina, two of these very highly enriched omega-3, in particular DHA tissues, and you can see there's differences from the Western diet through the fish oil to the fish oil with the PC, the phosphatidylcholine, whereas the saturated fats and the N6 fatty acids were higher in the Western diet group versus the N3s being higher in the other two groups. And then if you look at the retina, it's very substantially different for DHA compared in these diets. So these diets, as you can see, were achieving what we hoped they would achieve in terms of the supplementation with the fish oil. And now what we're doing with this particular data is teasing out how much of this was actually present in the tag form versus phospholipid form. And then we're tightening up this fatty acid distribution data as well. In terms of the metabolic disease risk factors, this is Jagathisan's part of the study. Interestingly, but it does match in with what those clinical trials were showing, there was no difference, no observed protective effect for fish oil supplementation or fish oil with phosphatidylcholine for any of our biomarkers, which included fasting blood glucose, total cholesterol, non-HDL cholesterol, fasting triglycerides. We did intravenous glucose tolerance tests, insulin sensitivity tests, and fat tolerance tests, and saw no benefit to the fish oil supplementation in any of these. So the next steps for our group are to get into the lipid species analysis. So determine specifically in that supplementation study how the PL, the phospholipid, the TAG, and the cholesterol esters varied between the supplementation groups. Then the next studies that we're going to be doing are looking at acute postprandial testing of specific fats and oils within a natural food matrix. So this would be looking at or comparing plant source omega-3s, so oil seeds that contain ALA or DHA, versus marine sources of omega-3s. And then we're also interested in looking at structured lipids. So using these plant-based or marine-based as the stocks, the feedstocks, to producing structured lipids. These will be interesterified lipids and fats where you can rearrange or custom design these fats to have fatty acids at specific points. And we want to look at that, how these are changing in the postprandial period. And then in addition to that, we would like to do stable isotope analysis, looking at dosing orally with specifically designed lipid species, in particular these structured lipids. So we can identify how these fatty acids are moving as they go from digestion through absorption in the enterocytes and then distribution into the other tissues. And in some cases, it's multi-stage. They might go to the liver first and get redistributed in the liver into a different lipid species and then end up in a tissue in a different form from which they were procured in the diet. So just closing up, in summary, there are still significant inconsistencies relating to supplemental omega-3 fatty acids in terms of their protective effects for metabolic diseases like coronary heart disease. These significant inconsistencies are important because we're not entirely sure how diet versus supplements should be used to benefit health. And what's needed, or the research gap, are very much more nuanced studies that need to focus on essential fatty acid ratios or total intakes of both linoleic acid, linoleic acid, alpha linolenic acid, as well as the EPA and DHA, and determine what the acute effects are and the long-term effects. These larger studies, where we just look at it in a single dose, massive population studies, they're not showing us what we need to see in terms of how these fatty acids can be used to improve health. And that's probably what the next steps should be from various research groups. So with that, acknowledgements, my co-PI on the study I showed you, Dr. Rob Bertolo, also from Memorial University, our graduate students who've been working on this, and as well some of that data was also provided by Hannah Murphy, very good undergraduate research assistant in my lab. And the funders for that data were Ocean Frontiers Institute and NSERC. Thank you very much.